For my citizen science project, I chose EyeWire. Okay, the formation of EyeWire. It was found by Sebastian Sung in 2012 out of Princeton University. Learning to use the software. Once I was on the platform, I was able to navigate through the different neurons, as shown above. These were some of the first tutorial neurons that I worked on. The blue shows the initial piece given to you, while the green shows what I got right, and the red shows what happens if you were to click on something besides the neuron. However, if you were to ever make a wrong move, it could easily be deleted and you could start working from the last correct place. If you look up in the top left-hand corner, that picture shows an example of me clicking on something wrong on purpose just to kind of show what would happen, and then you can just deselect that very easily. In the middle is a picture of a tutorial neuron after I completed it. The green shows everything that I got correct. There's no red and no yellow, which means I didn't get anything wrong and I didn't miss anything throughout this tutorial. And then on the right, that shows a tutorial in progress, but later in the tutorial, the green bar on the bottom shows how much of the neuron I've completed. So that neuron was almost completed. The blue shows the original piece that was given to you, and the green is everything that I clicked and added on. You can kind of see on the neuron how it's kind of encompassed by like a gray bubble. That kind of helped showed where the neuron path went. Staying inside the bubble helped to make sure that the neuron was whole and you weren't getting any incorrect pieces. Finishing the practice and moving on to the neuron cube. So these show my progress after working through the tutorial and beginning to move on to practice neurons and eventually to the real cubes. The image on the right and the left show it was no longer red and green, but a light blue as I got closer to the real cubes. So instead of it coming up as green when you got it right and red when you got it wrong, everything showed up blue. But as you can see in the left-hand picture, that progress bar is still there to kind of help guide you. And then as you see on the right-hand side, that progress bar disappears and you kind of just have to go off what you've learned. The image in the center is of my first cube, and the green shows what bits I got correct, whereas the yellow strip shows what was missing and the red shows what's wrong. So once you moved on to the actual cubes, it wouldn't tell you what you got right or wrong until the end. I was originally really nervous about getting things wrong because I didn't want to impact science in a negative way. However, so many people complete these cubes before they're ever accepted as full neuron bits and sections that you don't really have to worry about that, because there's also scientists constantly on the platform going through and fixing errors and discrepancies and making sure that everything is looking good. Why work on this project? Through this project, users can help to map the brain. This research and these discoveries can help provide doctors with new knowledge about different diseases that are associated with the brain. And once they exactly know how things work, it can help develop treatments and cures for patients that haven't been possible until now. On the right, you can see a picture of 6 different neurons. Through EyeWire so far, they have discovered 6 new type of neurons that had previously not been discovered. And all of these different discoveries and research that's being pulled is right now focusing on the retina part of the brain, but eventually they hope to map the entire brain. And just like personally and myself, like this is such a cool project because it can help to further science, further treatment, further cures for all of these brain disorders, diseases, and issues connected to the brain that we have previously known nothing about. So hopefully throughout the next several years, people continue to find new neurons, continue to map the brain, and it can benefit science in a possible way.